It's my pleasure to introduce Dr. Kevin Anderson. Dr. Anderson serves the Creation Research Society as the director of the Van Andel Creation Research Center. Kevin and I have known each other for a number of years, and yet when I asked him to come, he still agreed to do so, so I appreciate that. Dr. Anderson's area of expertise is in molecular biochemistry. Is that close enough?
Close enough.
Close enough, okay. But he's going to share with you, I think, some really interesting insights here. Without further ado, I'm going to turn this over to Dr. Anderson.
Thank you, Gary. And yes, we don't hold it against you, even if you are a computer person. So, up here, trying to figure out the computer. I always love that. Thinking, wow, yeah, computer's getting more complicated. Why? I don't know. Of course, my cell phone is still just the flip open, so. and the airport had to search it. They said, "Will you check your electronics?" I said, "What, my watch?" I did. Like, what are we talking about? Oh, well. All right. It is a pleasure to be here. Certainly when Gary invited me, I knew I had big shoes to fill because, of course, you had Danny and Dawn before. I mean, it doesn't get any bigger than that, although we're going to switch gears now. We're going to talk about something interesting, as like biology as opposed to stars. Okay, genetic insight into human origins. Gary, even though this is Origin of Life, you want me to talk about genetics. And so we will spend some time talking about genetics in this session and my next session, and then we're going to spend some time actually talking about genetics in my breakout session, which is actually called Rethinking Paleontology. In my breakout session on Wednesday, I'm going to challenge the whole paleontology paradigm from the point of a geneticist. And that was started because I was at a conference one time where a paleontologist was asked a question by a molecular biologist, and before it was done, as far as I was concerned, the paleontologist was flat embarrassed. And that got me thinking about things, and here we have it. It'll culminate in the breakout session then on Wednesday. All right. This kind of spins off a little bit of what Gary was talking about. So many times this whole issue is couched in terms of the Bible versus science, faith versus science, religion versus science. And you're almost presented with the dilemma that you have to choose one or the other, which I find rather silly, obviously, but certainly the secular society, the secularists, love that idea because then it stems, modern, analytical, objective, technical science versus that ancient, worn-out, repudiated thing called the Bible. That's what they want you to think in terms of. That's clearly not what this is about, and you'll many times hear that it's science says. Science says. Now, I've been a scientist for many decades. White coat, well, actually, I hate white coats, but anyway, laboratory bench scientist for many decades, and I have never heard science say anything. In fact, if it did, it'd probably scare me. Science is a tool. Scientists say a lot of things, but they're human beings. So next time you hear that science says, just read in there, a human being with limited understanding who will stand before God someday in judgment, says. That's what it actually is. Another interesting thing about this false dilemma is I was just a week ago, in fact, right at this time, presenting at an Assemblies of God conference in Springfield. And at that conference, they tried to be, shall we say, big tent. And they had a number of different views in there. They had theistic evolutionists. They had old Earth creationists as well as young Earth creationists. And one thing I found intriguing was consistently the theistic evolutionists and the old Earth creationists were always, how can we work to settle this dilemma between science and the Bible? Between what modern science says and what the Bible says. How can we work to settle this dilemma? And the young Earth creationists are all, there is no dilemma. Have you noticed that? We're the only ones who say there is no dilemma. There's nothing to settle. They're both totally true as long as you understand what science is actually telling us versus the interpretation by secular-minded people who are separated from God and will at some point in time stand before God in judgment. Okay? As long as you understand the difference, then there is nothing science says that contradicts Scripture. All right, so we recognize the popular view is evolution. That's a given. I don't think that any of us would say, oh, we think that if you're in the scientific community, that creation is the popular view. I don't think any of us are that delusional. Okay? Evolution is the popular view. Now, let's stop and think about it in terms of the popular view evolution versus what the Bible teaches. And let's understand what the Bible teaches and make sure that this is not our Bible, which is becoming very common. And I heard a lot of preaching out of this Bible last week from people. But in actuality, you know, this is what your Bible ought to look like. Does your Bible look like that? That's what it ought to look like. Because more than anything else, we ought to be in it all the time. Not relying on what our preacher says or what some college professor says, as smart as they may be, you know, but going into the Word and finding for ourselves. Because, you know, when you stand before Christ, you'll give an accounting for yourself, not for what someone else said. Can you imagine? You know, how does it feel? How does it sound when your child comes to you and says, well, they made me do it? You don't buy that. You think, is God going to buy that either? Okay? So you need to get in the Word. You need to study the Word. It needs to be your understanding. Between you and God, build a relationship. And you need to literally wear God's Word out in the printed form. Now, this is what I call the face. If you're a scientist, and you're working in the scientific community, and they find out that you're a creationist, you get the face. What in the world kind of nuthead, special kind of stupid are you? But we've got to recognize that that's what you're going to receive in a fallen world. In fact, would you expect anything less in a fallen world? Right? Would you expect anything less in a fallen world? That's a challenge that Christians need to think about. That's a challenge that Christians at this conference I was at a week ago need to seriously think about. Instead of being concerned about, well, this is the popular idea, or this is what is popular today, or this is what the majority say. In fact, what is it about the majority? If you went into a scientific conference, say the Conference of Cell Biology or Astrophysics Conference, and you said, how many here believe in evolution versus creation? And just at a show of hands, what would you expect? What would you expect? In a fallen world, what would you expect? Okay? We have to remember, we're not here to represent the popular opinion. We're not here to try to kowtow to the popular opinion because it's not based upon that. Truth has never been a popular vote. I cannot understand why Christians seem to think they need to worry about popular. In fact, what did Christ say? If the world hates you, remember, it hated me first. In fact, I would surmise if the world doesn't hate you, you're obviously doing something wrong. And remember also what Christ said, wide is the gate, and many will travel on it that lead to destruction. It's not a popular view. It's never intended to be a popular view. And Christianity and science, either one, should not be concerned about popular. They should be concerned about seeking the truth, understanding what is correct. Hey, let's talk for a moment about the contemporary view of human origins. It's obviously Darwinian. Okay, what that means is that at some point, about 12 to 16 million years ago, we shared a common ancestor with all contemporary primates. And one of those branched off and then evolved into the current gibbon. Then we have the 6 to 8 million years ago, we shared a common ancestor with the gorilla. And then about 4 to 6 million years ago, we shared a common ancestor with the chimpanzee. And this would be the contemporary popular view. If you opened up virtually any biology textbook, any paleontology textbook, any geology textbook, you'd see some kind of depiction of this. Okay, so for the last 6 million years, then give or take, because they keep kind of changing the numbers, trying to determine, but we'll say 6 million years, since our divergence from the chimpanzee human common ancestor, then human evolution supposedly followed some kind of path that looks like this. And notice we got bigger and taller and straighter as we went along. course, now we're starting to hunch over again as we stand behind our computers. Okay, so what I want to spend some time talking about is this versus genetics. Now, at a conference a number of years ago, Ken Ham was introducing things, and he was talking about we're going to take back astronomy, we're going to take back biology, we're going to take back genetics. And I contradicted him a little bit. He thought I was disagreeing, but I really wasn't. I said, we're not taking back genetics because we never lost genetics, did we, Jerry? We've never lost genetics. The evolutionists have tried to make some runs at it, but they've gotten slapped back every time. We've never lost genetics. If there's any field that evolution is terrified of, it's genetics. And boy, it is a fun time to be a geneticist and a fun time to be a creation geneticist. All right, for many of you, this word genetics and the concepts and the teaching genetics probably read kind of like this. Like, I have no idea what you're talking about. And I apologize for that. Kind of like my students, the eyes glaze over and they kind of just, look at you and everything. I will attempt as best I can to at least interpret some of this as we're going along, so you'll have a basic understanding of some of the genetic concepts. I can't promise I can interpret all of it for you, because it is a scientific field. It's a sometimes fairly rigorous and complicated scientific field. But we will try to keep it at least at a basic level that most of you can follow it, and it won't be too confusing normally. All right. Let's start out talking about the DNA in your cells. Okay? You have in your cells 22 chromosomes that are called autosomal chromosomes. You have a pair of each. You have 2 copies of chromosome 1, 2 copies of chromosome 12, 2 copies of chromosome 22. You get 1 pair from mom and 1 pair from dad. Where they came from. That's why you have 2 copies of each. Those are the autosomal chromosomes. And you inherit those equally, whether you're male or female. It has nothing to do with whether you're male or female. But it does have a lot to do with you genetically. Then there is another set of chromosomes. Those are the sex chromosomes, the X and the Y. You get 1 from mom, 1 from dad. If you have 2 X's, you're female. If you have an X and a Y, you're male. Okay. All of this together, then, is what we would call the human genome. So we're, in essence, talking about the DNA that makes up your chromosomes. Okay. A number of years ago, they began a project called the Human Genome Project, and the idea of the Human Genome Project was to sequence all of the chromosomes. Get the DNA sequence of chromosome 1, of chromosome 10, of the X chromosomes, of the Y chromosomes, and have it all mapped out so we can then go in and see, here's the DNA sequences of each chromosome. So the idea was then to be able to genetically tell what you are, okay, besides being sugar and spice. In 2001, the first draft of that sequence was then published. Co-jointly or jointly published in both the journal Science and Nature. Now, interestingly, at that time, the evolutionists really started making a lot of clamor about how the genome information just completely nails the door shut on creation. In fact, it was so strong that I was at a Gordon conference soon after the genome information was published, and I was talking to the individual that was the director of the Department of Energy's genomic project, and he said that there were many congressmen in Washington that were very concerned about the publication of the genomic information because it was so damaging to that stupid biblical idea of creation that their constituents weren't going to like it. And they were very concerned about that. So I grilled him some a little bit because he had no idea who he was talking to. I grilled him some a little bit of what it was that was going to be so damaging about it. And that's what a little bit I'm going to talk to you here this afternoon about, is what was supposedly going to be so damaging. Now, what's interesting is that with this initial hoopla about how wonderful the genome sequence information was in support of evolution and how this just nailed the door shut on creation, and those biblical creationists are going to have to just shut up because they got nothing left. I think it's very easy to say that never transpired, and in fact, it's been just the opposite. I'm really surprised, personally, that the evolutionists keep looking at it. I think they would want to just close it down and say, we're done. Because every day, it's just another nail in the evolutionary coffin. There's so many nails in that coffin now. I think you're running out of space to put another nail. And it's only getting worse, and we'll see just a little bit of it this afternoon. All right, so by evolutionary standards, the closer you are evolutionarily, in other words, the more recent your common ancestor, then the more genetically similar you should be. That would make sense, right? The sooner it's been, or the more recent it's been that you shared a common evolutionary ancestor, then there's less time for there to be genetic divergence between you and the other organisms. So, for example, we should be much more genetically similar to the chimpanzee than we would be to the ape, as example. In fact, to the point then that the chimpanzee was quickly then sequenced in 2005 because they wanted to be able to begin to do these comparisons. Now, what's interesting, and we'll talk a little bit as we go on in this, what's interesting is that the 2005 sequence published was really not a complete or a very well done. It's like they were kind of rushing to get it done. And they've had to go back and keep putting patchwork in then to try to fill in some holes from the original draft. But the claim has been made, in fact, this claim was made back in the 1970s, that we and chimpanzees share 98.5% similarity in our DNA, which would be consistent with an evolutionary perspective that, see, that's our most recent common ancestor is shared between we and the chimpanzees. So there's been not enough time for there to be very much divergence between us and chimpanzees, and therefore we're almost chimpanzee. Or maybe chimpanzees almost human, depending on how you want to look at it. The sequencing of both the human and the chimpanzee was supposed to solidify this and verify. In fact, we are 98.5% similar. Okay, let's look a little bit at some of the data. All right, if you go into the University of California Santa Cruz website, they are one of the curators of the human genome sequences. And if you go to their website, and if we were to look and analyze some of the, I don't know, is that me? We're getting some feedback here. Okay, do I need to stand away from something? I need to stand right here. All right, if we were to look at how the comparisons are made, and certainly the way it's presented to the public, you would get the idea that when they talk about 98.5% similar, okay, in your genome versus the chimpanzee genome, then that would mean, for example, that human chromosome number 1 and chimpanzee chromosome number 1 are placed together and they see the alignments, right? In fact, if you go on to YouTube and you look up a little short video by Richard Dawkins, that's exactly what he's doing. He's on a big board. He's going through, here's a difference, here's a difference, and it makes it look like there's hardly any differences, then just line them all up nice and fancy like. That's total fabrication. That's not how it's done. Here's how it's done. See, if you actually did it that way, and we compared chromosome 1 of chimpanzee and chromosome 1 of human, there'd be no comparisons. It wouldn't line up. And they know that. There'd be virtually 0% similarity. So, the way they do it, well, okay, I'm sorry. Then what happens is that instead what they have to do is they have to piecemeal the chimpanzee genome to align it to the human. So, for example, see, you take human chromosome 1. You don't compare chimpanzee chromosome 1. You compare small segments of chimpanzee chromosome 1 and chimpanzee chromosome 2 and chimpanzee chromosome 3 and 4 and 5 and 6. And so what we see is the different colors are different chromosomes. So you see you got the red and the blue and the green and the orange. Those are all representing different chromosomes that the pieces of DNA came from for the alignment. Okay? Let me illustrate that more so. In other words, let's say this is a sequence on human chromosome 1. Okay? What we do is we'd find a short sequence on chimpanzee chromosome 1 that would line up there, and then maybe a short sequence from chimpanzee chromosome 2 that would line up there, and then maybe a short sequence from chimpanzee chromosome 10 that would line up there, and a short sequence from chimpanzee chromosome 12 that would line up there. That's how it's done. And you think, wow, doing that, I think it's amazing it's even just 98.5%. I think you could almost make it 100%, you'd think. But it's even more than that. Because, let's take these two sequences in the box, okay? There's two differences. But depending on how you write your paradigm, or your protocol, and depending on how tight, how strict you want it to be, you can allow for a little bit of difference. So, for example, the T and the C and the G and the A don't match, but the rest of it matches. That's good enough, so we call the whole thing a match. Or we may have something like this. These are called indels, where it either is an insertion in the assumed to be either an insertion in the chimpanzee genome or a deletion in the human genome. Obviously, they used to be the same, okay, but now one of them is different. But what we do is we just simply don't count that. So this is considered a match because an indel we don't count. So it's even less stringent than what you were even thinking when we were just matching pieces to pieces. It's pieces to pieces with a lot of sloppiness even within the pieces. So depending on how tight or how stringent you write your program, you can match virtually anything up and call it a match. And they do. So this brings us to how is a genome even sequenced to begin with? And why is this the case? Well, first off, when you sequence a chromosome, like human chromosome 1. Human chromosome 1 is several hundred million nucleotides in size. You don't just feed that chromosome into a sequencing machine and out comes the sequence. It's too big. We don't have the technology yet. Maybe at some point, maybe Mr. Spock will have the technology, but we don't yet have that technology. So we have to sequence it in small segments and then line them up. And that creates some confusion. Also, we're not sequencing from a single human. First off, we have both an X and a Y. Well, obviously, if you've got both an X and a Y, or two X's, you're using both a male and a female. Okay? But I think more important for us, remember the human genome sequences were published in 2001, and the chimpanzee in 2005. Now, in order to kind of rush through and do the chimpanzee one a little faster, they took the human genome as a scaffold and built the chimpanzee one on top. Because, after all, we're virtually the same anyway. So, in other words, they introduced a heavy evolutionary bias just putting the chimpanzee genome together. Now, this in itself ought to disprove the whole notion because the 2005 alignment is recognized as no good. And yet, if it really was that we and chimpanzees were that close, it should have been a wonderful alignment. It's recognized it's not any good, but they haven't been able to figure out a better alignment. So they're still struggling to put the chimpanzee one together. And even with that heavy bias, where you use the human to build the chimpanzee, you use it as the scaffold to build the sequence of the chimpanzee, you still have, for example, human chromosome 1, we have, that would appear to be probably about 20%. Even after everything we just talked about, all of the fudge factors, all of the sloppiness, all of the tricks of the trade, you have about 20% of human chromosome number 1 that there is nothing in any of the chimpanzee chromosomes that matches. Now, human chromosome number 1 is your biggest chromosome. And if about 20% of it doesn't match anything in the chimpanzee genome, then it's a little hard already with just the first chromosome to see how you're going to get only 1.5% difference. If we look at human chromosome 9, not your largest chromosome, but not your smallest chromosome, and this is closer to about 25 to 30%. There's simply no match. No matter where we look on the chimpanzee genome, no matter how sloppy we try to make the comparisons, no matter how much we try to eliminate material that is, maybe not quite matching, how much we try to eliminate those indels and such, we still, 25 to 30%, there's still nothing. Still a huge gap. And then, of course, the favorite one of mine is the human Y chromosome. Over 50% of the Y chromosome in a human being has nothing whatsoever anywhere in the chimpanzee that matches. So I know, ladies, you've assumed that your husband was a chimp. Okay? But he might be a pig, but he's not a chimp. Right? Because there's so little matching between the human Y chromosome and the chimpanzee Y chromosome, even with all these fudge factors we've been accounting for. So the way the evolutionists explain the Y chromosome is they assume that in that 6 million years since we and chimpanzees diverted from our common ancestor, there's been massive mutation in the Y chromosome, but now it's stable. Isn't that amazing? Now it's stable. But it didn't used to be, but now it is. So, of course, in that sense, okay, if it's stable now, how do we know it used to not be stable? Well, it used to not be stable because if it has always been stable, then we have a real problem. So they have to assume it's not been stable. There's been massive mutation, even though it's stable now. And who's reading into the data, imposing interpretation, and who's looking at the data just as it exists? Okay. Also, you have to understand that even when they're making these alignments, taking pieces from any of the chimpanzee chromosomes, in a 2007 paper, for example, that was explaining some of the methodology, the programs they're using for comparison are very heavy biased toward evolutionary interpretation. That's an admission. It's assuming that chimpanzees and humans had a common ancestor. That's what's in the program, that assumption. Right? If there are repeats, and in the human chromosome, there's a lot of repeats. The computer doesn't know how to deal with those. When I say repeats, I mean the same sequence repeated over and over and over and over again. The computer doesn't know how to deal with those very well. And those don't work very well for these kind of comparisons. So what do? We just ignore them. Because they don't give us the kind of data we want, the result we want. So we just ignore them. They're there. In fact, we're finding out that they're very important to be there. But they don't give us the matching we want. And they're just useless endless repeats of the same sequence of DNA, so we just ignore it. As I've already mentioned, there's a heavy influence that we have a phylogenetic relationship to chimpanzee. And that's in the program. Any gaps or missing data, as we mentioned, if there is something where the human genome doesn't have it, or the chimpanzee genome doesn't have it, it's just ignored. And even with all of these biases, this paper said that at best, all we can get is 87.5% alignment. Did you read that in the New York Times? Nope, didn't. Okay, but let's look at another interesting little feature. Okay. We go into what's called the ensemble assembly. This is a group that work to develop assemblies of the human chromosome. And they reported, October of last year, that the human genome had a total length of 3.2 gigabytes. That's 3.2 billion bases. In comparison, in June of last year, right about a year ago, they reported the chimpanzee genome as having a size of 3.35 gigabytes. So if we do a comparison just using their data, we find out that the chimp is 7.5% larger than ours. That's even using the human genome, remember, to build the chimpanzee, and it still doesn't match up. Now, how can we be 98.5% similar if the chimpanzee is 7.5% larger? Well, remember, one of the tricks of the trade is if it's what they consider insertions, in other words, areas that the human doesn't have, what do? They delete it. See, so this 7.5%, it's not important. You just dump it. Now, the chimpanzee may not agree that's not important, but it's not used as part of the comparison. Interestingly enough, 4 months later, in October, the ensemble group, same data, but they redid some things, and guess what? They don't even report chimp genome size anymore. Now, they report what's called base pairs and golden path. Those are alignment data, but they're not genomic length. They don't even report the length anymore. And I just can't help but feel that one of the reasons perhaps is because they're tired of those, crazy creationists using their data. So they just don't report it. Just simply dumped it. And I'm glad I did screen captures so I can have what they used to report, because it's gone now. Hey, let's turn to another group. This is NIH's website. NIH, remember, is one of the groups that actually, this is your tax money at work, actually was involved in the sequencing of the human genome. They report the human genome to have a length of 2.8 gigabytes, 2.8 billion. Okay? The chimpanzee, then in November they actually reported a number of them, which I found kind of interesting, because how can you align the chimpanzee to the human if they're still trying to get the human aligned? So they have now a number of possible alignments and lengths of the human genome, all the way from 2.8 to 3.2. Okay? The chimpanzee, on the NIH site, they reported in June of last year as 3.3, and then just to show in November, they still were keeping it at 3.3. So the chimpanzee didn't change. It did on the ensemble, but not on the NIH site. So you see already the two sites aren't even agreeing with each other. And this gives us then a range. If the human is 2.8 to 3.2 and the chimpanzee is 3.3, then we have a range somewhere between 2.7 and 14.5% difference in the size of the two genomes, just depending on which human sequence you want to pick. But what this demonstrates is that we cannot, with any credibility whatsoever, declare that the human and chimpanzee are 98.5% similar, even though that's still found in the textbooks. And yes, I've found it even in 2014 biology textbooks, right in there. It's still in the textbook. It's still being what's taught to biology students in college, still being what you're going to read in the newspapers and such. But you see, there's obviously no credibility whatsoever to that argument, just based on this, even if you didn't know anything else. Okay? Now, Svante Pääbo, he was early on a very adamant proponent of the 98.5% similarity. But even by 2007, he had become disenchanted enough that he said, quote, "I do not think there is any way to calculate the number." And in the end, interesting, it's a political and social and cultural thing about how we see our differences. In other words, it's not a scientific, it's political, social, and cultural. So if your worldview, Gary was talking about worldviews, if your worldview is that you and primates, you and chimpanzees share a common evolutionary history, then you will be likely to assume 98.5% difference, not based on science, but based on your worldview. Okay, let's change gears for just a second and move into actually looking at DNA itself. The chromosome is composed of DNA. Hopefully we're not going to get too much Chinese going here, but it's composed of DNA. DNA is composed then of nucleotides. They contain sugars, phosphates, and what we call nitrogenous bases. The nitrogenous bases are adenine, thymine, guanine, cytosine. Now, the backbone of the DNA, the sugars and the phosphates, never change. It's always the same whether it's bacteria or human. That doesn't change. That's like the piece of white paper. Right? That's always the same. What changes is the sequence of the nitrogenous bases, simply just the bases, A, T, G, C. So when you see DNA sequences reported, you generally see them reported in a fashion of just writing the letters, A, T, C, G. This would appear to give us a linear code, not unlike English. In fact, this becomes very attractive to look at the DNA sequencing as a linear code, just like we would look at English as a linear code. You have letters, 4 letters, A, T, C, G. You have words, which we call in genetics codons. You have sentences, which, for example, would be what we refer to as open reading frames. You have punctuations, which could be, for example, things such as promoters and stop codons. And you have paragraphs, which we may refer to as genes. See, so there's very much a linear thought to this, just as you would think of English as a linear written communication. In fact, when I took genetics a few years ago, It was very emphasized, the linearity of the DNA code, the linearity of genetic activity. We have to understand this because this mindset has driven a lot of the interpretation of genetics in the last 50 years. Okay? Walter Gilbert, Nobel Prize winner in 1992. Didn't have the human genome results yet. Okay, 1992, referring to the size of the genome, said that in computer terms it's actually very little. 3 billion bases of a sequence can be put on a single compact disk, and one will be able to pull a CD out of one's pocket and say, here's a human being. It's me. See the linearity in the thinking. Why humans are so simple, we can put you on a compact disk. Put you in a computer, pop you back up. That was the mindset. There's a human being. Everything about you is all right here on this CD. Okay, well, when that drives then the thinking, then you begin to think in terms such as junk DNA. Junk DNA is your evolutionary leftovers in the DNA world. Okay? What that means is that it appeared that some 98% of the human genome did not code for protein. And the thinking was if it doesn't code for protein, then it doesn't do anything, so it must be junk. But you see, this is very heavily based on what I call that linear code mentality. Protein has to be the product. If that DNA is not involved in making protein, it must not be doing anything, therefore it's junk. It's useless. Okay? So what's that mean from an evolutionary perspective? Well, see, they would say, for example, that you have the fish. The fish has the gene for fish gills. But now, as it evolves, and it's no longer a fish, it's now walking on land, it doesn't need those gills anymore because it's not breathing oxygen out of water, it's breathing oxygen out of the air. I need to get back over here. Okay? So it doesn't need that gene for fish gills anymore. Well, it doesn't just chop it out and throw it away. That's not how DNA works. But what happens is because you no longer need the gene for fish gills, that DNA now no longer serves a need for that organism. And like all DNA, mutations can begin to enter in, and they change the sequence. And so after a while, the sequence begins to become more random. But it doesn't hurt the organism because it doesn't need that DNA anyway. And so by the time you're walking around and looking like your husband last weekend, that gene that was essential for the fish to make its gills now is just random gobbledygook sequences in the human DNA. Parts or all of it are still there, but it doesn't serve a purpose. Hence, it's junk. And the assumption was that the vast majority, 98% perhaps even, of your DNA is just this junk. In 2006, Francis Collins wrote in his book, The Language of God. If you're not aware of Francis Collins, he was the director of the Human Genome Project at NIH. He then has subsequently become the director of NIH. But he's also become a Christian, but he is a very outspoken evolutionist and very critical of what we're talking about here. Okay? In his Language of God in 2006, he said a certain amount of hubris was required for anyone to call any part of the genome junk, given our level of ignorance. And I would clearly agree with that. You don't understand how the genome is working, how can you say this piece doesn't have any function to it? And yet, just a few pages later, he says in the same book, Darwin's theory predicts that mutations that do not affect function, namely those located where? In the junk DNA, will accumulate steadily over time. And this is exactly what is observed. See? So even though it takes hubris to call it junk DNA, I guess he feels he's got the hubris, because here we go. And in fact, he continues on. Junk DNA is exactly what Darwin's theory would predict if, as some might argue, these genomes were created by individual acts of special creation. Why would this particular feature appear? In his Language of God, junk DNA is a very significant part of his argument against creation, even though it takes hubris to talk about it. In 2013, on an atheist website, talking about junk DNA, they said that junk DNA only makes sense if this DNA was inherited from a common ancestor. Homologies between junk DNA are probably the most powerful of the homology evidence for common descent, as common descent is the only rational explanation for them. In other words, what they're saying is that we and chimpanzees would have the same junk in the same area. The only way that could come about is if we both descended from the same common ancestor. But this is making one very classic assumption, and that's what? I don't know, do I have to wait the full 7 seconds? Okay. Yeah. Yeah. I'd never wait 7 seconds on my students. They'd be asleep by then. So anyway. Okay. What's this making an assumption about? It's making the assumption that this junk is indeed junk and doesn't have a function. In other words, it's making that hubris statement that Francis Collins mentioned. Okay. In 2007, a consortium of researchers called the Encode Project analyzed 1% of the human genome. Now, you'd say 1% doesn't sound like very much, but if you think we're talking about over 3 billion nucleotides, 3 billion letters, see, 1% is going to be what? Can you quickly do the math? And you don't have your calculator in front of you, right? And they looked at it in great depth. Not just cursory look in the sequence, but they looked at it in great depth. What's going on? What's it doing? What's happening in this area of the DNA? And they were looking very heavily at some regions that were considered junk. And they found that most of the area they were looking at, in fact, up to 93%, actually was being transcribed. In other words, there was activity being done and making RNA from it, and they called this, in their own words, unexpected. Why was it unexpected? See, because they were evolutionists. They were assuming it wouldn't be doing anything. Right? Following that, then, in the journal Nature Review Genetics, the statement was made that these observations suggest that genomic architecture is not collinear. Remember we were talking about that linearity? They're saying, we're looking at this in more detail, and guess what? It's not linear. Instead, it's interweaved and it's modular. And the same genomic sequences are multifunctional. Now, we don't have time, really, this afternoon to get into multifunctionality very much. But just think of it in terms of if on a page in front of you, that word "the" was used in several different ways for several different purposes in reading that page. That gets very complicated very quickly, and that's what we find in the genome. Multifunctionality of sequences. Not just a single linearity where that one word has one meaning in that one sentence. It has several meanings in multiple sentences. Okay? Now, since then, the Encode Project has come along with an additional study in 2012, and they've published several papers since then following up on this and finding even more activity, even more detail to your genome. And this led then to the observation that the work by the Encode group supports and is consistent with earlier observations of a highly interleaved transcribed genome. But more importantly, prompts the reconsideration of the definition of a gene. When I teach my biology students today, I have to tell them, we'll talk in terms of gene, but we don't know what a gene actually is anymore. I don't know how else to explain it, so we'll still use the archaic term, but that's what it is, an archaic term. We no longer understand what a gene is because we're no longer looking at just a linear code. And there'll come a time, maybe in some of the young men up here's lives, they'll see it, where the term gene will be thrown out in place of something else because of a better understanding of what it is. Gene is based on linear code, but genetic activity is not linear code. In fact, we have to move beyond the linear code to really understand genetics. DNA is a three-dimensional molecule. I don't know if you watched Carl Sagan's movie Contact, where they got the information from the UFO people, and it was written in a three-dimensional code, and they were just all so enamored and wowed by that. But we already have that. It's in your chromosomes, three-dimensional code. But here's the key. It's read in a four-dimensional dynamic. You know what I mean by that? What's the fourth dimension? Time. Your chromosome changes constantly, and every change it makes changes the genetic activity going on in your cell. Gary, how would you like to write a computer program that was three-dimensional but read in four-dimensional dynamic? Now, here's the challenge. And we'll challenge our computer guy here. Yeah, here's the challenge. Okay? Could you read that code, three-dimensional, read four-dimensionally? Yeah. Could you write it? See, here's the challenge. 3-dimensional, read 4-dimensionally. And yet, that exists. That exists. It's beyond us, but it exists. It's in your chromosomes. It's already been written. That book that we wouldn't know how to do exists. Okay? Much of the junk, what was called junk, is used to make non-coding RNA. Originally, this is assumed to be transcriptional nonsense from these dead genes. In other words, it's residual leftover from when you used to be a fish, when you used to be an amphibian, when you used to be a frog, when you used to be a lizard. And it's still there in various pieces, see, so it's leftover nonsense from these dead genes. But instead, now that we've understood it better, we're beginning to realize more and more that instead, that stuff is not dead genes. It's not residual leftover from your evolutionary past. It's very active and very important, particularly in areas of regulation, in areas of making the genetics work the way they're supposed to work, creating that 4-dimensionality, creating that where it's going to do one thing right now, something else 5 seconds from now, and something else different 10 seconds from now. Okay? The structure of the chromosome itself is vitally important. You change the chromosome structure, you change the genetic activity. If you fold a page in a textbook, does that change what's written on the page? No. But it changes what's written on your DNA. Okay? Regulates gene expression, enables or blocks replication, and allows for that 4-dimensional activity of the chromosome. Okay? There are on your DNA segments called enhancers. Now, the enhancer may interact with a gene. Again, we don't have a better word, so we have to use gene. Okay? It may interact with a gene, but that gene may be thousands of bases away. So you've got to actually bring them around, so you've got to fold the DNA just right for that to happen. If we change all the sequence of the DNA between the enhancer and gene 3, we change how it folds and it no longer works. So can we have junk in between the enhancer and gene 3? No. Here's one called chromosome kissing. You didn't know chromosomes were rheumatic. 2 different chromosomes, chromosome 1 and chromosome 7, may decide then to bring 2 portions of their chromosomes together. It's called chromosome kissing. Once again, see, if we change the sequences on either of those chromosomes, we can't achieve that. So the sequence itself is vital to allow this kind of 4-dimensional dynamic of the chromosome. Paper that just came out, 193 new proteins found from regions that were thought to be non-coding. In other words, they do not have the standard gene structure to them. Hence, one of the reasons we don't know what a gene is anymore. Okay? In fact, these were thought to be, for the most part, junk. So now we have to recognize when we look at the DNA, we can no longer say this is a gene, this is not a gene, because we don't know what genes look like anymore. 193 unaccounted for. So 193 new genes that don't fit the standard definition of a gene. And 404 proteins that come from what is called long non-coding RNA, which is supposed to be RNA that doesn't do anything, but it's making protein. 404 of them making protein. How? We don't know, because we don't understand, because it doesn't fit the standard understanding of how that works. So here's papers that just came out about 5 weeks ago, showing more of the linearity thinking. The linear code doesn't work. There's activity going on that we now are only beginning to tap into that is beyond the linear. It's the 3-dimensional with the 4-dimensional activity. And here's some of the first absolute proof of that. Okay? So let's use an analogy. We talked some about a computer program. Let's maybe put it in terms of a book, assuming you've read a book before. Okay? I use that with my students, and they're kind of like, no. But anyway. All right. We'll say it's analogous to a 2,000-page volume of War and Peace. I would sometimes say Pride and Prejudice, but you know what Mark Twain said about Pride and Prejudice? He said, if any library that does not contain Pride and Prejudice, even if it doesn't contain any of their books, if it doesn't contain Pride and Prejudice, it's a good library. So I guess he didn't like Pride and Prejudice. So I'll use War and Peace. All right, so 2,000 printed volume of War and Peace. But if it was written the way that your chromosomes are written, it would be on 20 pages. And we're not talking about 20 pages of very, very small print. We're talking about 20 pages of normal print. But you read it in such a way that you read it the standard way, left to right, top to bottom, turn it over, left to right, top to bottom, fold the pages, read it again, fold the page, read it again, fold another page, read it again, bring a portion of another page, put it into with that page, read it again, and you keep doing that over and over because you've got to read this book three-dimensionally, but it's got to change all the time, see that four-dimensional aspect, and you got it all in 20 pages. And then this is back to the question that I asked Gary with the computer program. Would you be able to write that book? Would you even know how to read that book? And yet, you see, that book has been written. It's your genome. And then when we get to junk DNA, we say, well, there's simply no room for junk. There's simply no room for it. Because the detail in your chromosomes, maintaining that three-dimensional structuring, acting in a four-dimensional dynamic, cannot have vast amounts of junk and still function. Just like our 20-page version of War and Peace can't have a lot of junk in it and still be able to function to serve, to give us the story of War and Peace on 20 pages. Now, hold the presses. Okay? According to Carl, my friend Carl, I ought to say he's my friend. He and I have battled back and forth enough over the years. Okay? According to Carl, we've known all along it was not junk. You didn't know that. It wasn't junk. No. In fact, apparently, now I'm not quoting him, I'm paraphrasing, but apparently, say they never really said it was junk. And if they said it was junk, then they really didn't mean it in the usual way that we mean junk. Okay? And if they did mean it in the usual way, then you misunderstood. They're really just playing a word game. Because they really didn't say it was junk. And if they did say it was junk, they really didn't mean it was junk in the usual word. So, yeah, exactly. See, so, wait, totally innocent. Oh, we didn't call it junk. No. But alas, their evolutionists have another problem. Quote, while junk is expected, a junk-free genome is not. Whoa, wait a minute. We've got to have some junk in here. That's not going to work for us. And here's the reason why. Estimates of the human mutation rate suggest that somewhere between 70 and 150 new mutations strike the genome of every baby. Okay? See, so your children have mutations. And of course, you already knew that. But your children have mutations in them that you don't have. But see, you have mutations in you that your parents didn't have. And it's just being passed on down generation to generation. More mutations, more mutations. John Sanford does an eloquent job of talking about this in his book, Genetic Entropy, which, by the way, we have for sale on the Creation Research book table. Okay? All right? Based on the risk of mutational meltdown, which is also called air catastrophe. In other words, you just build up so many mutations that just finally the organism just dies because it's got too great a mutational load in its chromosomes. Okay? Based on the risk of mutational meltdown, it is estimated that only 10% of the human genome can be functional. In other words, we need that 90% junk as a buffer to put all the mutations in. See, that's what they've been relying on for years. Where do you explain these mutations going? Well, they go into the junk. Okay? The other 90% must be junk. Emphasize, must be junk. Why? Because if a mutation alters junk DNA, it doesn't do any harm because the junk isn't doing us any good to begin with. So it doesn't make any difference. But if our genome was 80% functional, the figure batted around when the Encode Project results first came out, then, what's he say? We should be extinct. Now, what I find ironic in this blog, he wrote this, by the way, in his National Geographic blog, he never answered the question of why we're not extinct. I felt like going on there. I didn't have time. I felt like going on there. I was like, Carl, old buddy, you haven't answered your question yet. Why aren't we extinct? Well, the assumption, of course, is that it's got to be junk. We cannot tolerate too much non-junk in our genome because then we've got no place to put the mutations. And evolution doesn't work. So the evolutionists don't want junk to go away. That's why I was kind of teasing Jerry. I don't know why they just don't stop the research. because every day there's less junk. There was a website that was monitoring how much junk was in the human genome, and they stopped in 2011. I think the reason they stopped is because it was not looking good for them. There's too much non-junk anymore. But really a bigger question is this. Because he's saying, well, does that mean I should be extinct too? Because the same applies to the primate. And so what we see, just in this very big overview to begin with, is the genetic support for human evolution is steadily going down the drain. And this is just the beginning. And we'll talk about more tomorrow. And so with that, I will close. My wife says it's a redundancy. Have 2 pictures of me up there. But I will, Gary said to go, we were told him to go ahead and promo, so I will promo my video, Testing Evolution. And I guess we're actually supposed to have a few minutes for question and answer.
Do you find moderators?
I can do that, yes. Yes?
What's your prediction? What percentage of DNA will be shown?
Boy, that's a tough one because it's so difficult. Let me give a little analogy as part of the answer. Genetics has been, over the course of the last several decades, genetics has been like we discovered King Tut's tube. And, kind of modest, but very interesting. A lot of gold, a lot of interesting treasures and such. And as we're going through mapping and we're logging everything and we're describing what we have and such, it's like we got to the far corner and somebody noticed a little crack in the wall and they kind of pushed on it and it opened up. And you notice there's a little hallway. And you kind of walk the little hallway and all of a sudden you come up and you realize that's just been the porch. And you come into a room that as far as you can see up and as far as you can see down and as far as you can see across is nothing but gold and treasures. And that's what we've done is we're just now standing looking at that, realizing we don't know anything anymore. So in answer to your question, I don't know. I mean, I would guess we would still say the human genome is going to contain some junk because we've just been degenerating. Even in the 6,000 years, give or take, since Adam and Eve, we've been degenerating. But it's certainly not going to be, more than just a few percentage. So I would think the trend certainly is to say every day there's something new coming on. Every day there's another discovery. This is now functional. This is now functional. You're just going to see the ticker just going, more and more function, more and more function. So I would say before we're done, we're going to be well over 90% would be my prediction. Yes?
What regulates the folding of the DNA?
Well, there's several things. One of the things that regulates folding is there's specific proteins that do that, and also there's RNA. So in a sense, you have to make RNA from the DNA to come back to make the RNA again. And that's kind of back to the chicken and egg, which evolutionists have a lot of trouble with, but creationists obviously don't. So you have both of those acting together that bind to proteins and bend and fold. Now, we've known that protein, for example, will bind and fold our DNA for a long time, but it hasn't really been appreciated the importance of that nearly as much as the last few years have shown. So the proteins interacting and some of the RNA interacting with the structures will help maintain the shape of the fold and even be what will determine, are we going to pull this piece of DNA out and do chromosome kissing or not? See, and it's the proteins controlling that. And the RNA sometimes controls that. The RNA may control the proteins in some instances.
I've always been curious about parts of
the histone is what the DNA, because the DNA is actually wrapped around histones, which are proteins, in order for you to pack the DNA in. Now, we didn't have time to go into that, but if you took, for example, in your cells, if you took and unwound all the DNA, I guess I need to get over here again. Okay. If you took and unwound the DNA in your chromosomes, it'd be, it would be a lot bigger than you. Well, you got to get all that into the inside of your cell, in fact, in the nucleus in your cell. So you have to pack it very tight. So part of that packing process is you wrap the DNA around those proteins called histones. But they're clearly involved in it because histones will be part of the shape. and they will interaction with the histone will be part of how you unwrap and rewrap the DNA. So they're clearly involved in it. And there's a lot of other events going on that I haven't even begun to discuss. Other question? Yes?
You talked about the mutation.
Mm-hmm.
What's observed? What's theorized?
It's not necessarily theorized for evolution to work, because it's not based upon we got to go have this much in order to account for the changes in humans since our common ancestors 6 million years ago. This is what is observed. In fact, I think evolutionists would assume that not be the case, because you see that's where they're having problems with the junk, because they got to account for if your genome is millions of years old, which it actually is. In fact, it'd be older than that, because your genome would trace back to the beginnings of life. Okay, but even starting with the chimpanzee moving forward, if your genome is 6 million years old, even in that case, you don't have the ability of absorbing 75 to 150 mutations every generation for the last 6 million years, and it not be total meltdown of your chromosome. So it is the actual observed number of mutations per generation. Yeah, way back there.
As the cost of the genome analysis decreases, when will it be possible that we should all have our genomes analyzed?
Well, already you can have parts of your genome, like the 23andMe. Yeah, the 23andMe. They're not going to sequence your entire genome, but they will sequence certain areas using markers to determine, do you have susceptibility to certain genetic diseases. So they're already doing that. But yeah, as the cost gets less and less, there is talk about at some point in time you will have your own personal genome on perhaps some kind of chip on your thumb. You go to the hospital and put it onto the scanner, and they'll bring up your entire genome. I just tell my students, everything about you genetically will be known, up to and including what genetic defects you have, and if you have herpes and all those things. It'll all be known. So probably at some point in time, how soon, who can say, because it's partly economics and partly technology. But it is getting cheaper enough that we are sequencing we're sequencing a lot faster for a lot less than it took to do the human genome. And that's why you're seeing more and more other organisms being sequenced now, because it is getting cheaper and is getting faster.
But it changes in time. So how do you know that?
Well, the sequence doesn't change in time. The structure, the shape changes in time. And then the key thing is, to take that sequence and predict the shape, that we can't do. So I wanted to make sure I'm clear on that. Yeah, we're not talking about the sequence changing in time. The shape changes in time. A change in your sequence, that's called a mutation. And you really probably don't want those. Yeah. We'll talk a little bit more about mutations tomorrow in my lecture tomorrow. Yeah?
Let me get to my question. We're sitting here in pharmacy school. Contemporary pharmacy education talks about gene-specific therapies for chronic disease. Are you positing then that ultimately isn't going to work? Or
I'm not saying it isn't going to work, because I think certainly there's some aspects of it will work, but I am saying that make sure I word this correctly, so far many of the directions, many of the paradigms have failed because they're very linear in their thinking. And when you would go into, for example, looking, and junk DNA is also part of that. They're only now looking, or only really making a concerted effort just in the last few years to look at junk DNA as involved in genetic diseases, which they hadn't been looking at for quite a while. They talk about creation being a science stopper. Here's an example where evolution was a science stopper. They should have been looking at those regions years ago, and only now saying, this region is actually, when a mutation happens, actually brings about this disease. So it's been slow in responding. And it also has been slow in trying to actually put the true story together, because sometimes you have great success and sometimes no success at all. And you don't know why. And well, one of the reasons why may be because sometimes you don't need the three-dimensional aspect, and sometimes you do. I mean, clearly with sickle cell anemia, that's just a one-point mutation. We know exactly what it causes. We have a good idea what's going on there. But other mutations causing other types of problems, we're not sure why that mutation is causing that problem.
chimps compare to similarity between chimps and gorillas or birds and reptiles?
Well, again, see, it's going to depend upon how you're wanting to approach it. It's going to depend essentially on how you work the program. With similarity between, let's go ahead and use similarity between chimpanzee, gorilla, and human. There's been some work where they've actually shown that in up to a quarter of our chromosomes, we're more similar to gorilla than we are to chimpanzee, which shouldn't be the case. So it just depends on how you're doing the comparisons and the paradigm you're using to do the comparisons. And I don't have the quote in here, but I have a quote where essentially they say that the human genome is just a hodgepodge. We can't determine an evolutionary lineage to it, which is what we would expect as creationists, or certainly is not inconsistent with creation, but it's very inconsistent with evolution. And that was a troubling concept for them, that we can't determine an evolutionary lineage. We should be able to, but we can't, because it's just too hodgepodge, just like that, where, okay, 25% of it's actually closer to gorilla. Oh, how'd that happen? Well, they got all kinds of hypotheses. Some of them are a little strange, okay, because they want to say, well, there was early periods of time where you're having some kind of horizontal transfer between human and gorilla. I don't even want to picture that one. But anyway, so, yes?
You indicated that the human genome study did not involve one individual person.
Correct. It was several people, right.
What happens if you compared?
Yeah. And that's part of the weakness, if you will, because every human has what are called SNPs. That's single nucleotide polymorphisms. In fact, if I go back to this slide very quickly, the reason they think they can discount the TC mismatch and the GA mismatch is because they call that a SNP. It's a single nucleotide polymorphism. Humans have that also, where if we compared your chromosome number 1 to someone else's chromosome number 1, there'd be these areas where they just simply, you'd have a T, they have a C. You have a G, they have an A. We don't know why. We're not sure exactly what's going on there. And that's every human has that. And so the reason when they compare chimpanzee and human, they assume the same thing. Well, that's just a SNP, don't worry about it. And so those aren't counted then as part of the differences. They're actually counted as part of similarity. So you have those kind of differences. And then, of course, you have other differences depending upon, what you inherited. You may have inherited, I don't know what color your eyes are versus your wife's eyes and such like that. You see, so you may have some of those differences too. By and large, between humans, there's going to be approximately 0.3 to 0.5% difference in your DNA, by and large. And that's also one of the things I always try to get my college students to understand. Genetically, there's no such thing as race. Genetically, there's no such thing as race, because there's no single genetic trait, no single genetic marker that is consistent or found in just one people group that identifies them versus everyone else. And the reason, again, is because there's really such small differences between us. Yeah?
Then what is the next step? What's the
Well, obviously that becomes the question, depending on the procedure you use for aligning. One of the problems we have, as I mentioned, is the chimpanzee chromosomes were put together, if you will. When they sequenced and then put all the pieces together, they used the human genome as the scaffold to build that. So they put in a huge evolutionary bias into the putting together, which is not working. No competent genomist recognizes the 2005 chimpanzee assembly is very good. But we don't really have a better one. There's a little better one they did in 2009, but it's still got a lot of problems to it. So the answer to the question really is we don't know. Except I think it's safe to say it's a lot more than 1.5%. And some of the work that Jeff Tompkins has done at ICR suggests it might be closer to 75%, 80%. So it certainly is a lot more different than 1.5%. And the paper I cited, even the 2007 paper, was themselves admitting it was over 10% different. And again, that doesn't disprove evolution, but it disproves the predictions they were making based upon their assumptions of similarity. But it's fully consistent with the creation model.
How would you explain this to fifth grade class of studying gems.
Well, I think the simplest way to explain it probably to a fifth grade class is probably the way you explain it to a college class. And that would simply be that you'd have to recognize that the way the comparisons are being done is not what you think they are. It's not chromosome 1 compared to chromosome 1. It's not chromosome 2 compared to chromosome 2. They have to take small sections and compare them together. So a lot of it is based strictly upon when you're writing the program, what built-in assumptions you put in the program. I know computer scientists don't ever make assumptions, but yeah, what built-in assumptions you put in the program. And so you're going to find what you assume you're going to find, is what it really comes down to. So if they want to find 98.5%, they can get it in the program to do that. But then it's very easy to challenge because you say, but you got this assumption and this assumption and this assumption. So if you write a program that is, shall we say, minimized in assumptions, then you're probably down to it's going to be 80 to 85% or less similarity. Yeah.
You said something in passing, and I don't want to let it slide. All too infrequently, the whole concept of race is passed over. Scripture, the only races are those who believe the promises of God and those who don't.
Yeah.
We belong to Adam and Eve. That's where we came from.
Right.
That is probably the one thing that has caused more problems than anything else in our culture from Darwin, is the idea that there are inferior races and there are superior races.
Now, it didn't,
Get that handled.
right. And, of course, Jerry's written a whole book about that. But in fairness to Darwin, he didn't start that. But he certainly poured gasoline on the fire. Yeah. Certainly poured gasoline on the fire. And, of course, part of the reason for that is because race, as it's commonly viewed, is very visual. Color difference and such. And so you can immediately see, well, they're different than I am, color-wise. But genetically, that color doesn't mean that much, genetically. And yeah.
So when you say that chimp genome in 2007 isn't working,
When they did, in 2005, when they were sequencing the chimpanzee chromosomes. Remember how they sequence chromosomes. You can't just put a chromosome in and get a sequence at the end. You only can do it in small sections. And you got to align them. Okay? How this, and many times you're going to have an overlap. You got to figure out where the overlap is. So they thought the fastest way to do that was they already have the human assembled. Although apparently now they don't, because we saw they're changing their ideas of the size of the human. But they had the human assembled, so the idea is, well, let's use the already assembled human. We know there's only about a 1.5 difference. So they were using the assumption, so let's just use the human to build the chimpanzee. So when we're trying to align, we'll say, oh, this aligns here, because that's how it is on human. So that's what I mean of what they did in 2005, but it very quickly became apparent that doesn't work. Because, okay, this assembly is not right. Thereby showing, just on itself, we are not just 1.5% different from chimpanzee, because if we were, that assembly would have worked beautiful. The simple fact that it doesn't work, the simple fact that it's well known it's not working, that things are not assembled correctly, that we don't have a proper assembly of the chimpanzee chromosomes is a strong evidence that we're not just 1.5% different. Because can you imagine saying we're going to take this organism that's 50% different and use it to align? Yeah, you'd say, well, that won't work. Exactly. And while we may not be 50% different, we're not just 1.5% different. And I think that's the evidence of why we're not aligning. One more question? I think you've been trying to get a question in.
Yeah, maybe. We're sitting here 6,000 years after planet did the side of that. How do you take that into account? What happened?
Well, it wouldn't have been right.
Fallen creation, we don't want to look at it.
Yeah.
We want to come to a perfect creation, what God did.
Right.
And so all of this that you're doing here is looking at a fallen creation.
Correct.
How do you take that into account?
Well, of course, and that's obviously part of the difficulty. Now, the changes that would have occurred in us genetically wouldn't have occurred all at once. And in fact, we feel that biblically it's consistent to say that a lot of it occurred right after the flood, because you see human lifespan dramatically dropping right after the flood. Well, something must have happened biologically. And we think one of the explanations for that is there was an acceleration, as the RATE project talks about an acceleration of radioactive activity, there was an acceleration of mutation. And so that could be an explanation for the rapid deterioration of our genome. So it wasn't an all at once thing from the moment of the fall. Suddenly your genome deteriorated. It'd be a slow process, but perhaps accelerating at the time of the flood. Now, how to get past that or back from that? See, that's not necessarily easy. We can do it in bits and pieces. Rob Carter, for example, studying mitochondrial DNA, which is DNA in another organelle in your cell called the mitochondria. It's not a very big piece of DNA. He thinks that he's gotten Eve's sequence within just 6 bases. See, so it's not impossible, but it's very difficult. Of course, chromosomes would be more difficult, it's so much bigger. So it'd take a lot of work. But there are people such as Rob Carter that are working on it, trying to go back and say, let's get closer to what it used to be. Let's get closer to what the original sequence used to be as way of trying to determine things such as our human lineage. But all that work shows, for example, that you can't do it evolutionarily. In other words, we can't go back to our common ancestor. And the evolutionists are admitting that. We can't take the genetics and go back to our common ancestor because we simply find too much, we're here all of a sudden we're closer to gorilla than we are to chimpanzee.
Theologian and as a
Mm-hmm.
I think you're putting too much importance. You're putting more importance on the flood. You did that. You're going to still have to go to the fall.
Sure.
And
Right. But the reason we look at the flood the way I said is because we see that dramatic drop in lifespan. So that would seem to indicate something biologically had happened.
that we should, I think,
Yeah. No, I'm not saying that everything stayed pristine up to that. That's obviously not. But that biblically gives us a point where we say it seems something really dramatically happened much faster than what had happened before. Because up till the flood, lifespans were still fairly consistently the same. And after the flood, they very rapidly dropped. And that is a biological change. One we can't prove right now, but one would assume that would have involved genetics as well.
Let's thank Kevin